Hi guys, today we're covering chapter 7, which covers photosynthesis. So basically the gist of photosynthesis is kind of understanding that everything on Earth is dependent on solar energy. Not that everything is able to photosynthesize, but we who cannot photosynthesize are very dependent on those who can. So all organisms that are able to use sunlight to store energy, essentially they're able to take sunlight and make sure that that energy can be utilized and transformed into the stored energy of carbohydrates. So that's the process called photosynthesis. Individuals who are autotrophic are able to produce their own food, and so photosynthesizers are autotrophic. Everyone else, like us, we have to consume our food, and so we are considered heterotrophs. Photosynthesis happens in the green portions of the plants, and so there are these specialized organelles called chloroplasts, and those are the things that are involved in photosynthesis. Those are the things that carry out photosynthesis. And the reason why they are green is because one of the most important pigments that plays a huge role in photosynthesis is chlorophyll. So it is a green photosynthetic pigment that essentially like attracts solar energy. And so what happens is those pigments bring in solar energy to the chloroplast, and so then the chloroplast can do its thing. The materials needed for photosynthesis are carbon dioxide and water. And so carbon dioxide actually diffuses in through the stomata, which are these like little small openings or pores that are in a leaf. Note that photosynthesis happens in all the green portions of the plants. So the leaves, that's where you can find all the chloroplasts. And so that's where photosynthesis is going to happen. Water gets, of course, taken in by the root system and then drawn up to the leaves so that it can be utilized for photosynthesis. So some chemical abbreviations that we need to know. We definitely need to know the abbreviation for glucose because we use it all the time. Oxygen, water, carbon dioxide. There are other important abbreviations here. We don't need to know both names there. We just need to know some of these important metabolites that you see here. So this is a huge component of photosynthesis, is understanding that photosynthesis is really divided in half. So there's the front portion and then there's the back portion of photosynthesis. The front portion of photosynthesis involves the light reactions. And so for the light reactions, they happen only in the light. So they're appropriately named. Science gets it right sometimes. And they are the energy capturing reactions. And so the light reactions are super critical for getting all the energy components that can be used to power the Calvin cycle reactions. So without the Calvin cycle reactions, you don't have energy. It's basically like getting in your vehicle and you didn't put gas in it, so that vehicle isn't going to go anywhere. Calvin cycle reactions aren't going to happen without those light reactions, okay? So the light reactions essentially, in this process, chlorophyll absorbs solar energy. That energy jazzes up electrons. The electrons will move down an electron transport chain, just like we saw in oxidation reduction reactions in the last chapter. That process pumps hydrogens inside this thylakoid space, and that is essentially used to make ATP out of ADP and a phosphate. And also one method of that process, one portion of that process also generates NADPH, which is a really important electron carrier that gets used in the Calvin cycle reactions. So all of the light reactions happen in the thylakoid, okay? Everything else happens, like the Calvin cycle reactions happen in a chloroplast, but it happens in the stroma, that gooey center of the chloroplast. And so with the Calvin cycle reactions, CO2 essentially gets reduced to a carbohydrate. Remember, reduction is gaining. Remember our oil rig. That's super important for these oxidation reduction reactions, is understanding that photosynthesis is an oxidation reduction reaction, or a redox reaction. So oxidation is losing, reduction is gaining. So there are some pretty important pigments that play a huge role in photosynthesis, and essentially those pigments are just chemicals that absorb certain wavelengths of light, and those that aren't absorbed get reflected back. And this is critical because light, visible light, happens on a spectrum. And so pigments found in chlorophyll absorb different portions of visible light. So it looks green because it absorbs a lot of the reds and the blues. There's another important photosynthetic pigment called the carotenoids, and those are essentially, they reflect these yellows and orange lights. So we're seeing the carotenoids right now remain when we're looking at these new fall colors that we're seeing. The super important photosynthetic pigments are chlorophylls A and chlorophyll B. So those are the major photosynthetic pigments in plants, and they absorb violet, blue, and red light. The carotenoids are an accessory pigment. They're also very important, and they essentially help pass energized electrons to chlorophyll A. There are also, these pigments also come together to form what are called photosystems. So these are super important, and we'll talk about those in just a bit. This is I love this image because it shows you the visible light spectrum, which we learned as a kid as Roy G. Biv, red, orange, yellow, green, blue, indigo, violet, right? And it shows you the role that chlorophyll A, chlorophyll B, and carotenoids play, and kind of where they appear in this light spectrum. So some important energy components, like the light reaction is all about generating energy resources to power the Calvin cycle, right? So important energy components of these include ATP, which is the energy currency of a cell. We learned that already. And we need ATP to reduce carbon dioxide to a carbohydrate. And then also an NADP plus, that's nicotinamide adenine dinucleotide phosphate, and that's a cofactor of a redox reaction. So remember, cofactors are kind of like little assistants for all these processes, and that is reduced to form NADH. So essentially that helps to carry electrons that are used to reduce CO2. So essentially the light reactions consist of two alternate electron pathways. So remember, an electron transport chain is just a series of proteins embedded in a membrane passing high energy electrons, boing, 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 down these proteins that are embedded in the membrane to a final electron acceptor. So here, the light reactions have two alternate electron pathways. So in the non-cyclic pathway, electrons bounce from one protein to another until the end receptor. In a cyclic pathway, those electrons get jazzed up in the first photosystem, and then they go kind of around in a circular kind of process from protein to protein, and then back to the same photosystem. And it looks a lot like, trying to show you my image here. So here's my photosystem. Here are my proteins, right? Here's the path that electrons could follow, or or it could follow this pathway where you've got photosystem 2, protein, 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 all the way down to photosystem 1. And then that electron would be jazzed up again in that photosystem and then passed here where NADPH could be made. Unfortunately, the photosystems were named by order of discovery, not so much by order that we see them in the thylakoid membrane. So the non-cyclic pathway that happens in the thylakoid membrane uses those two photosystems. And remember, a photosystem is like a big bunch of pigments that kind of act as like an antenna to absorb light. So you'll see photosystem 1 and photosystem 2, but not in that order. It's photosystem 2 first that captures light energy. And then you see photosystem 1. So essentially, the electron will get jazzed up in photosystem 2 and then travel down the electron transport chain to photosystem 1. And that continues, that process continues because water is there, right? Water is a resource we need for photosynthesis to happen. And so water gets split, and then the electron that's hanging out associated with water gets passed down that pathway. Oxygen then diffuses out. Those hydrogens will work from splitting water, and they're collecting in the thylakoid space right here. That hydrogen that's building up inside that thylakoid space is going to be like very high amount of hydrogen. It's going to be tons of hydrogen packed in that space. They're going to want to go from high concentration to low concentration. So photosystem 2 is essentially that first photosystem, that first pigment complex that you encounter in the thylakoid membrane, and it is the thing that's getting electrons from the splitting of water. Oxygen then diffuses out as gas. Those electrons that get energized in that first photosystem get passed down from one protein to another that's embedded in that membrane, that thylakoid membrane, and passes those electrons all the way down to photosystem 1. At the same time, hydrogens are pumping into this thylakoid space from the stroma. So you can see here, there's photosystem 2. You can see hydrogens are accumulating in that thylakoid space, and some of those are coming from the stroma. Some of those are accumulating here from the splitting of water, because we need to keep feeding electrons to go down this transport chain. Then an electron will go to photosystem 1, which jazzes that electron again, and photosystem 1 will then have that electron being passed from that photosystem to what's known as an NADP reductase enzyme, and that enzyme actually allows NADPH to form here. So an electron will pass from photosystem 1 all the way to this enzyme, which then attaches that hydrogen on there. Another important complex to note is the ATP synthase complex. So that's also associated with the thylakoid membrane. And remember that we built up a ton of hydrogens in that thylakoid space. They're going to want to go from areas of high concentration to low concentration. And so with that, they're going to want to go out one place, and that's through that ATP synthase complex. And so essentially, and that's a term called chemiosmosis. And so it has all these hydrogens flowing out this one little enzyme, right? And that hydrogen flow causes osmotic pressure. That pressure actually brings together ADP and a phosphate so that ATP can be made. Okay, so here's my take on hydrogen flow out that enzyme, right? And bringing together ADP and a phosphate, so you get ATP. This is a little bit nicer drawing than my little marker drawn image of the thylakoid membrane, but it works. So we talked about how hydrogens build up in that space, and that's a really fancy word called chemiosmosis. It's literally just osmosis of a chemical, because you build up all those hydrogens in that space. They can only go out that ATP synthase enzyme, and that allows ATP to be created. The next and very important step to the process of photosynthesis is the Calvin cycle. So we've had the light reactions. They've generated that reaction has generated all the energy that we need to power the Calvin cycle, and that actually occurs in a series of reactions right after the light reaction. So essentially, carbon dioxide from the air is used to produce a carbohydrate, and it's divided into 3 steps. So there's carbon dioxide fixation, carbon dioxide reduction, and then RUBP generation, which will make much more sense after we talk about this. This is kind of an important image that you'll find. Note that the Calvin cycle happens in the stroma, not the thylakoid membrane, and it is a cycle, okay? So because you know that it is a cycle, we have to talk a little bit about how many times this cycle has to turn just to produce one important metabolite. The first thing that has to happen is that you have to take atmospheric carbon process, like this only carbon source you have is carbon dioxide, because you want to make a carbohydrate. And so you do that by taking carbon dioxide from the air, and you essentially attach it to a 5-carbon sugar called RUBP, or ribulose biphosphate. Okay, so RUBP is now attached to carbon dioxide. That allows you to have a 6-carbon molecule, which then splits into 2 3-carbon molecules. And of course, like most good processes are, this reaction is accelerated by an enzyme called rubisco, because we've taken carbon dioxide from the air and attached it to RUBP. Now, and then we broke it down into 2 3-carbon molecules. We've now we've attached or fixed our carbon source. So now we can do something with it. Now we want to reduce it. Remember, reduction is gaining. So we want to take that carbon source that we got from the air, and we want to whip up a carbohydrate. So we've got to gain some stuff here. So that 3PG that we made, remember we had a 6-carbon molecule, we broke it up into 2 3-carbon molecules. We reduced that to BPG, and we reduced that to G3P. Essentially, it's called glyceraldehyde 3-phosphate. I like to call it PGAL because it's just a really fun name for this. And also we've got all these 3's and these G's and these P's, and it just gets really confusing here. So we call it glyceraldehyde 3-phosphate PGAL, and this is a carbon skeleton that stores energy and can form larger molecules like glucose. We need PGAL to make glucose, fructose, sucrose, galactose, all the oses that are important carbohydrates for plants. They're all built on the backbone of these PGAL molecules. Essentially, this process involves using NADPH that we made in the light reactions and ATP as well. We need 3 RUBP molecules to converted to make 6 PGAL molecules, but that is a little tricky because we can't just leave our Calvin cycle empty. Like we have to be able to we have to cycle it again so that we can have some RUBP left in the system to attach to incoming carbon sources, right? So the last step then becomes RUBP regeneration. So RUBP has to be replaced because we can't just leave, like we don't just come home from a trip and leave our gas tanks completely on empty so that we can't go another trip the next day, if that makes sense. So we got to regenerate RUBP so that we can start all over again when we have carbon again. So for every 3 turns of the Calvin cycle, we need 5 p-gals to make 3 RUBP molecules. So obviously the Calvin cycle is super important because that p-gal molecule that we're making can be converted into lots of other molecules. It can be used to make fatty acids and glycerol. It can be used to make glucose, fructose, starch and cellulose, which we definitely need, and even amino acids. So the product of photosynthesis obviously can be converted into many different resources. Like we know cellulose is great for cell walls. We can make more RUBP. We can use it for energy and use it for, of course, flowers and seeds. Plants also have the ability to store these carbohydrates. So they did all this work of making those carbohydrates, and usually they have leaves, roots, or stems that are adapted for food storage, because you don't want to make up all your carbohydrates and then not have a place to go. So roots are a great place for carbohydrate storage. Stems, some stems are modified for food storage, like potatoes or, different things like that, any kind of tubers. Leaves, some plants have like leaves that have like swollen underground leaves called bulbs, right? And those are great for food storage. We know those as onions. Even daffodils can do it. The process that we talked about is C3 photosynthesis. And so many types of plants won't perform C3 photosynthesis. Interestingly enough, there are other types of photosynthesis. So the normal process, Rubisco is used to fix CO2 to RUBP in the mesophyll cells. But this gets it's not possible to do in hot, dry climates. So what happens in a hot, dry climate, what plants have to do is close their stomata to avoid the issue of water loss, because when water is lost, then they'll wilt, right? But we know that those stomata have to open to let CO2 in. And so what happens is the stomata close because water is important. We got to save water. But then we don't get CO2 into our system. And so CO2 decreases, oxygen increases. Oxygen, kind of as an emergency, starts combining with RUBP. That can lead to the production of CO2, but it's really not efficient. It's actually called photorespiration. It's super common, and it happens a lot in C3 plants. Other types of photosynthesis, some plants are able to do what's called C4 photosynthesis, and those are known as C4 plants. And so they can avoid photorespiration altogether because they use a different sugar and a different kind of structure here. So what C4 plants do is fix CO2 to what's called a PEP molecule or a C3 molecule. And the result is it makes oxaloacetate or C4 molecule. And so that actually allows Rubisco to always have a high CO2 concentration. In hot, dry climates or when temperatures go up, C4 plants can avoid photorespiration, and their net productivity is like 2 to 3 times higher than C3 plants. So like corn is a really great example of a C4 plant because it does just fine when it's hot and dry. But in like a cool, moist environment, C4 plants can't really compete with C3 plants. They do it way better because they don't have that extra step. This just shows you like a chloroplast distribution in a C4 plant versus a C3 plant. And this shows you an example of a C3 plant and a C4 plant, right? Another type of photosynthesis is CAM photosynthesis. And so that's called crassulacean acid metabolism. CAM plants are really cool because they're able to partition carbon fixation by time. And so at night, when it's cool and not so sketchy, the sun isn't out, it's not so crazy, CAM plants are fixing CO2, right? They form C4 molecules, they're stored in large vacuoles. So then in the daytime, the light reactions can happen. NAD, pH, and ATP are available. They leave their stomata closed for water conservation. And so all that fixation that they did at night of CO2 then gets released so that this whole photosynthesis process can continue. So a pineapple is a really good example of a CAM plant. Obviously, each type of photosynthesis has its advantages. C4 plants are most adapted to high light, high temperatures, limited rainfall. C3 plants are better adapted to cool, moist temperatures. CAM plants can really survive in extremely arid places.